everyone. My name is Anjali Gujral, and I will be moderating the event today, along with my colleagues, Jitin Varghese, Mark Hutchison, and two of our students, Amna Soniwalia and Sonia Kim. So first, I'd just like to thank you all for joining us today for the Emory Global Diabetes Research Center Symposium, which we've entitled Diabetes Endotypes, the Way Forward. And we have organized this as part of the National Diabetes Awareness Month. And we thought it was timely to host this symposium as the thinking around diabetes development and treatment is changing, and it's no longer a one-size-fits-all approach. Advances in biomedical research have identified what have been called subtypes of type 2 diabetes, or what we are calling diabetes endotypes. And what this means is that different people develop diabetes in different ways. And this can lead to diverse presentations at diagnosis, as well as risk for complications. And so these different endotypes have implications for how we think about prevention in individuals, how we think about response to treatments, how we think about risk of progression to complications. Again, as I mentioned, it's no longer a one-size-fits-all approach. But this novel research is showing that there are actually differences between these endotypes or between these subgroups. And with that, we believe that knowledge and detailing of these endotypes can better characterize the diversity that clinicians have known about for some time, but it's important to consider where we go from here. So therefore, we've assembled a panel of experts on the topic from around the globe who will review current evidence on diabetes endotypes, as well as share their vision for the future of research and novel clinical applications for precision medicine. So in the first hour, we've divided the event into 2 hours. And so in the first hour, we will have the pleasure of hearing from Doctors Norbert Stefan and Miriam Udler, who will present reviews about what is currently known about diabetes endotypes and discuss ways forward. And in the second hour, we'll hear from a panel of experts who I'll introduce at that time, who will discuss their visions for the future of diabetes endotypes. And before we begin, I'd just like to thank the organizing committee for all of their work in putting this symposium together, comprising of members of the EDGRC faculty, staff, research assistants, and students. So with that, I would like to now introduce Dr. Stefan from Helmholtz University Munich in Germany, and he will discuss a review of history and current knowledge of endotypes and thoughts on the way forward. So please go ahead, Dr. Stefan, whenever you.
Thank you very much for the introduction. And I share my screen, and please let me know if something wouldn't work. I hope you can see my screen now.
Yes.
Okay. Yes. Dear colleagues, I thank the organizers very much for the invitation to participate in this interesting symposium, online symposium about diabetes endotypes. And I want to share a little bit with you my view, so how they could be used more from a clinical practical aspect. I'm based in Tübingen, Germany. I'm a clinical scientist, so I mostly work 80% with patients in the field of diabetes research, but also cardiovascular research. And yeah, let's get started. I think I make a little introduction because I'm the first person to speak today about this topic. I want to briefly give you the background about why it's so relevant to talk about diabetes. Just because we know diabetes is a leading cause of death these days, but it will be a much higher leading cause of death, or more importantly, cause of death in the years to come. And that's not trivial. And regarding our patients, but also politics, it's very important to see how the diagnosis of diabetes compares to other diseases in this field of research. So the next question, also very important, new data indicate is diabetes problem because of years of life lost, and most of you certainly know that that's the case. So if a person, for example, in this 60s develops diabetes, female or male loses about 4 years of age. If the diabetes has been diagnosed in this person 10 years before, then with 50 years of 15, then this person loses about 6 years of age. And so you see the increase, meaning the early diabetes diagnosis is being is there, the more years of life loss. So it's a very relevant disease, and certainly we're trying to understand what are the most important causes for the disease and the complications. Another important figure is this one. It projects the age standardized prevalence of type 1 and type 2 diabetes until 2050. And you see this is our global data, the steep increase in the prevalence rate that will come in the next decades. And it's also important already thinking about a little bit about subtypes or endotypes, that we know that there are large differences in the prevalence. And for example, in North Africa, Middle East, there's a much higher prevalence rate of diabetes compared to other countries. And we look for the high income countries and Latin America, Caribbean countries, they actually have the same prevalence rate. So there is already a heterogeneity there appearing where people meaning I hope that I'm still there. Can you still see my slide?
No, we've lost your slides.
Okay, so for some unknown reason, I need to. Okay, do you see it now?
Yes.
Okay, sorry.
Dr. Stefan, could you please hide the floating meeting controls so we're not able to see the titles of your slides?
So what should I hide?
The floating meeting controls.
Floating meeting control. Oh,
yeah, this is perfect. Thank you.
Is okay, better. Okay, sorry.
Yes, better.
Okay, so another aspect is that, for example, here projected for white populations with a BMI of 30, you can see the different ethnic backgrounds of diabetes incidence rates, meaning that Black people, Chinese, Arab, or South Asian ethnic background develop diabetes at much lower BMI. So already we have there a heterogeneity in the diagnosis. So we know that the pathogenesis of type 2 diabetes is very complex. We're talking about genetic risk, about the unhealthy diet, and also the sedentary lifestyle. But then we know it's a very heterogeneous pathophysiology, meaning some people most probably get obese, and they get diabetes because of a heavy or large obesity risk they have. Others mostly get diabetes because sorry, for some reason, this really makes a problem today. Okay, so I hope you hear me. And
yes, we do.
Okay, sir. And so certainly at the very end, the question is when people get insulin resistant, so moving from right to left, those can compensate with higher insulin secretion. They don't have actually, don't get diabetes, don't get hyperglycemia, but those who drop off the line become pre-diabetic and develop type 2 diabetes. So you most probably all know these studies. I just briefly introduced it, MIPIS and Life Group put this data nicely together, looking on subtypes of diabetes and focusing on age of onset, HbA1c, BMI, insulin resistant, insulin secretion based on fasting measures, and glutamate deoxyribonuclease antibodies. So when they took a very conservative approach, you divide their group in patients with type 1 diabetes and autoimmune diabetes, LADA. You see the prevalence as you expect. But when they did this clustering based on these parameters, they've identified a different picture, meaning that there are clusters. For example, this diabetes, severe autoimmune diabetes, reflecting the type 1 diabetes, and then insulin deficient diabetes, insulin resistant diabetes, mild obesity-related diabetes, and mild age-related diabetes. So the most prominent clusters, as I said, is the more type 1-like diabetes with GAD antibodies and impaired insulin secretion. The insulin deficient diabetes are often leaner people who actually have a moderate insulin resistance but strong impaired insulin secretion, and those with severe insulin resistant diabetes, particularly having high BMI and insulin resistance. But also when you look on this graph, you see for HbA1c, BMI, age, and so on, the different clusters, but you can also appreciate the large variability that is there. And certainly the models is not perfect. So certainly it is with this very hard clustering, you can not actually very well separate the people. So they just get or they are just being put in a certain category. Now, working with the clusters, certainly they found that for nephropathy, people with the severe insulin-resistant diabetes, the green line, they have a very, very high risk. You can see the mild obesity-related diabetes compared to that, not a lower risk, and the others in between. For retinopathy, it appears that particularly the severe autoimmune and insulin deficient diabetes resulting in early hyperglycemia is more of the driving force. And for coronary artery diseases, again, it is more the insulin resistant group that has the highest risk. But there is not that much of differentiation compared to nephropathy. So how do these clusters behave in other studies? And here is the origin study. And here in this study, people early on got insulin, basal insulin, and you can see those the clusters help to separate, like the glargine was more effective in the severe autoimmune diabetes or the severe insulin deficient diabetes, no difference in the insulin resistant diabetes, and again a little a separating effect in the mild obesity related diabetes. So it appears that for insulin resistance, with treatment with basal insulin, it is not important to knowing whether you are in the one or other cluster, but for the insulin deficient cluster, certainly it is helpful to identify these clusters. Now the question is novelty always new? So do we have in hand already other ways of separating people and defining different endotypes? And Dennis will speak afterwards, and I hope I correctly present this data briefly. Also did the analysis in the ADOPT and RECORD study, and he and his colleagues nicely found or recapitulated the different clusters proposed by Altwist and Life Group. And when they looked on the progression of HbA1c, so the increase over time, there is a separation. The clusters defined, so those here in the mild obesity cluster have a strong increase in HbA1c, others not so much. But when they took age at diagnosis, they could actually see that the cluster didn't have much of an improved prediction. So with a diagnosis of age at diabetes onset, you actually can recapitulate and project what will happen in these people. And similar for decline in renal function, It was also the clusters somehow separate, so people who actually decrease in GFR or increase in albuminuria. But if I understood correctly, and John just tell me if I'm wrong, the correction for GFR at baseline already predicted a similar outcome. So the classes were not more helpful of separating people regarding their incidence of renal impairment. There are some interesting findings about treatment in these studies, that particularly in the insulin-resistant cluster, thiazolidinediones, the insulin sensitizing drug, was more effective than the other ones, and appears a little bit that in the mild age-related diabetes cluster, sulfonylureas, that normally you see the strong increase in HbA1c, so you don't want to give them to insulin-deficient people, appears not to be that of a detrimental effect regarding HbA1c change. So they have some little predictive effect in the choosing of the pharmacological treatment of type 2 diabetes. Now, trying to give a little bit of a big picture is a summary of the clusters. So the ALKS clusters, as I've reported here, and Miriam Udler will talk about her approach to clusters, so I don't go in detail. And so also we know that there are like soft clustering, meaning where you have the opportunity not to put people in hard clusters, but where you use some continuous prediction where people are more related to the one group or the other group. All have some advantages and limitations, and mostly the limitations are in my view, that the prediction of outcomes is relatively poor at the moment. So we hope that this cluster will perform better. Now, again, what other approaches could be used, or maybe some combination of clusters and other phenotypes, as I call. So we know also in prediabetes, early on, that we can somehow separate people, for example, normal weighted prediabetes, so the skinny people, prediabetes, you have the high proportion of those who only have insulin secretion failure. So they don't have insulin resistance, don't have fatty liver, don't have visceral obesity, but certainly it is not that discriminative if you see how much overlap is in the other, like 50% of people. But it is some approach of taking phenotypes to enrich this cluster. So this is some idea for the future. And another concept that I'm working on with my colleagues is we call metabolic health. So it's a little bit of an elaborative change compared to metabolic syndrome, but I just want to tell you this not rocket science. So people thought about this way of stratifying people for cardiometabolic list since many years. So what does this mean? So metabolic health means that the older definition is that people have less than 2 risk factors of the metabolic syndrome not accounted for waist circumference. And when I look in my data, there are, for example, the lean people, and red, those are metabolically unhealthy, the blue are metabolic healthy, and here are the prevalences of the at-risk phenotypes. And interestingly, the strongest difference in the lean people is that if they have a low percentage leg fat mass, like the gluteofemoral area, then they have a high prevalence of metabolic unhealthy condition, and not so much in the overweight and obese group, so you don't have the stratification there. So in like principal components analysis, following up on the high sub-Q leg fat mass percentage, you see that it's, again, we use the term clusters in a phenotype where we have high insulin secretion, high sensitivity, high cardiovascular fitness. So it appears that this stratification using phenotypes may help to advance a little bit this very rough and hard clustering that we did before. And here are summary data about metabolic health. So people either took the metabolic syndrome or less than 2 risk factors of the metabolic syndrome, not accounted for waist circumference in most cases. And you see those obese people who are metabolically healthy, they still have an increased cardiometabolic risk. But look, on the lean people, their risk is really extremely high when they are metabolically unhealthy. So we were not completely happy. So these are meta-analysis. So it's that more than 1,000 publications about this concept of metabolic healthy obesity. And then we went further and said we need to take into account some aspect of body fat distribution, as I have shown you before, that we think that the lower body fat mass is very important and protective. And we came up with a new definition, and we tested it in the Enhance study UK Biobank. And if we have a new definition, we see that actually there is no increased risk. So we have still about 40% of the obese people in here, but it appears that with this newer definition that was empirically derived and then tested in other populations, it may be protective. So what I want to message is here that we still have accepted risk factors like blood pressure, diabetes, yes or no, but importantly, a body fat distribution aspect. And also, even if we take this metabolic health aspect, we still have a large variability. So in those people who we assume to be metabolic healthy, we still have this high waist to hip ratio, we still have hypertension. So we are definitely not at the end in defining novel risk stratification models. And again, summarizing it a little bit, giving an overview. So I think the metabolic health is very good for risk prediction, for risk communication, but it doesn't give you any information about pathomechanisms, a little about treatment. And then the diabetes cluster, like ARCWIS cluster, has I think it's very good for risk communication, not much of pathomechanism. The genetic-based clusters, Miriam will talk about, is very good for pathomechanisms in my view. So, and the other cluster analysis, like the tree method Ewan Pearson developed, I think are very attractive and new aspects, and hopefully we can use it to better allocate people in a more of a pathomechanistic risk group. So I just want to give you the last draw attention to a very important publication where a large group of people working on precision medicine, and it's absolutely worth reading it. And there are many aspects of going much deeper in risk stratification, of diagnosis, of prediction, of treatment aspects, how we can better define subgroups, endotypes of people with diabetes. So I would like to come to a conclusion. So in my view, I think the concept of metabolic health and cardiometabolic risk clusters have attracted definitely much interest. So many people are really interested in following up on what we are talking about today. I think the metabolic health concept is easily used because widely available clinical variables are in most cases used, predominantly metabolic syndrome, but also estimates of insulin-resistant subclinical information and body fat distributions, and in most cases, established cutoffs can be used. The cluster approaches so far are also based on routinely available parameters, but also can include more complex data, just like we call omics, particularly genetics, metabolomics, and other omics. So regarding cardiometabolic risk stratification, I think both the concept of metabolic health and cluster are so far not considered superior to establish risk prediction models in the general population. So we have diabetes, CVD risk prediction models like the SCORE2 and so on. So they are really good for individual prediction of the risk. However, they may be helpful for better predict risk in subgroups. So we talk about the lean people, the very obese people. We talk to people in prediabetes. So it may be that here the risk stratification by cluster or metabolic health may be important. And we think that in the future, cluster approaches will maybe used to allocate people better to pathophysiologic risk groups. So I think this is really the large benefit of using this endotypes, because here we can now really talk about the major pathophysiologic risks that lead to disease, in this case of type 2 diabetes. So with that, I'm at the end, and I thank you for your attention.
Great, thank you. Thank you so much, Dr. Stefan, for taking the time to share with us today. So we have a few minutes for questions. If anybody has questions for Dr. Stefan, we've enabled the question and answer feature down below. So if you have a question, please type it into the question and answer box, and then I will go ahead and moderate and ask Dr. Stefan. So please feel free to ask questions if you have any. Waiting for a minute. I don't see any questions yet.
It was a little bumpy ride with my presentation, sorry.
No, yeah, that sometimes can happen with virtual presentations. So that's the downside. But the beauty is we can all be together from all over the world and don't have to leave our homes, even if we don't want to. Okay, I don't see any questions yet. Okay, I have a question from an anonymous attendee. Is the metabolic health-based cluster, or in the metabolic health-based cluster, what are the prospective findings you observed using soft cluster approach compared to hard cluster?
Oh, that's a good point. And actually, I didn't do that, but this is on the plan. So looking particularly and do a pre-separation like the metabolic health. So established, you can take the metabolic syndrome as well, and then do the soft clustering. I think that would be important, and yeah, important question, but I haven't done it yet.
Any other questions? Oh, okay. Now they're coming in. All right, here we go. Okay, so we have another anonymous attendee saying, thank you for summarizing the diabetes phenotypes clusters. Have you seen the same for obesity, and would it be worth doing?
For obesity, I see. So I personally didn't do the clustering for obesity, but a colleague of mine, Robert Wagner, did a clustering in people with at risk of type 2 diabetes. And he came up with 6 clusters, and so certainly we have the lean ones in there, but also overweight and obese. So the clusters appear to be similar to the Alpes cluster in the diabetes, but so far we haven't done really specifically looking on the lean and on the obese. It's because that would be the important question. So just to summarize, if we move from the diabetes to the diabetes risk cluster without diabetes, we see a similar stratification, but it's absolutely worth the question, and that's a good point. It may really be, and it somehow alludes to the first question, whether when we do sub-stratification by BMI, that's certainly easily done, we may find that the different clusters evolve. Similar as I've shown you in our 2017 publication when I looked on the phenotypes, so the specified phenotypes, they differ quite in the normal weight compared to the overweight and obese people. Yeah.
Okay, and we have a question from Dr. Voss. It looked like visceral adiposity and fatty liver were clustering together. Is that correct?
If they're clustering together, no, actually. Let's say different. In most cases, they do. But I haven't prepared this data, so we published in 2023 a paper cell metabolism, where I tried to separate this. So I think we can separate visceral obesity from fatty liver, and that's really tough doing that when we take into account the most important fatty liver produced hormones, hepatokines as I call them, and adipokines. So unfortunately, I didn't present this data, but I think we can do this very tough job of separating visceral obesity from fatty liver in determining insulin resistance. And interesting question.
Okay, so we have two more questions. So the first one is from Tim Mori. So on your last slide, you talked about BMI metabolic health stratification in subgroups and how these cluster approaches might improve on established prediction models. But then couldn't we simply derive a classic prediction model in these subgroups as opposed to clusters?
Yes, and I think most people didn't do that. So they use classic risk prediction for type 2 diabetes, for CVD, and so on. But very seldom people in epidemiological study looked on this pre-stratification. And I think that's really worth doing, and it's so easy to do that, to really do this sub-stratification with BMI at first, and then look at the prediction. So we know already that, for example, for type 2 diabetes prediction, they don't work that well in the more obese people. And they work well in the leaner, but not in the obese people. But this is something actually that should be easily be done, and proposed a modification of this simple risk stratification based on things we have already, like BMI, body fat distribution, etc.
Okay, so last question is from Dr. Narayan. So most of the data driving these conclusions are from well-nourished, high-income white populations. The paradigm is also driven by insulin resistance as the insulin resistance as the dominant theme. What if type 2 diabetes is a problem of the islet cells? Are we missing something?
The last, I didn't answer. If it is a problem of the
islet cells or the beta cells.
Okay, I understand. Absolutely correct. And that's the point that we always need to take into account. The clusters always work with the parameters you put in. So if we don't have good measurements of insulin secretion, we completely miss insulin secretion. We have good measurements of sensitivity. And so therefore, not only certain sensitivity is a very important driver, but I think in most cases people miss the insulin secretion defect just because it's so difficult to precisely measure it.
Right. Okay. So thank you again, Dr. Stefan. We really appreciate you taking the time. And so now we are going to hear from Dr. Miriam Udler from Harvard University as well. She's at Harvard and at Mass General Hospital in Boston, Massachusetts. And she will review the current evidence surrounding type 2 diabetes endotypes from a genetic and clinical translational perspective, as well as, again, giving her thoughts on the way forward. So Dr. Udler, if you're ready, please go ahead.
Great, thanks. Can you see my slides okay?
Yes.
Okay, and actually I'm going to move the top part, make sure, let's see if that's better. Okay, how's that? Good view. Okay, great. Well, thank you so much to the organizers for inviting me to be part of this really exciting webinar. And I think the topic is really important and interesting, and I'm excited also to hear the panel this afternoon. And Dr. Stefan gave such a great overview. That was fantastic. And so this, hopefully what I'll be speaking about will be a nice segue from what he just presented. So I'm going to focus on specifically what we can learn about diabetes endotypes from genetics. Okay. So, just as we all know, in adults, over 90% of diabetes is type 2, and we deal with this issue that it's a diagnosis based on a very non-specific endpoint of high blood sugar. And in fact, with type 2 diabetes, we essentially have a diagnosis of exclusion where we say someone doesn't have autoimmune diabetes or other forms of diabetes that we can pinpoint. And so that's, really what's motivating our desire to subclassify. And the hope is that by subclassifying, we can improve management for patients. So in preparing for today, I was thinking back about the concept of endotype, and it's a term that's thrown around a lot. And so I look back at one of the first manuscripts that introduced it. And so it's from the asthma literature, so a paper back from 2011. And the way that they defined it there was that it was proposed to be a subtype of a condition defined by a distinct pathophysiological mechanism. So I think when this is very appealing, I think, to all of us, because the idea, and that what they go on to say in this manuscript as well, is that the dream is that if you can identify a distinct pathophysiological mechanism, then you can target that for treatment, and so therefore improve treatment for people with the disease. And genetics is very well suited to help us dive into pathophysiological mechanisms. So I'm going to just take a moment and just give kind of an overview of how I think about genetics and diabetes. And this is a schema that's often used, where on one axis, we see the different frequencies of genetic variants that can contribute to disease. So on one end, we have very, very rare variants that may only be seen in one family in the world, that contribute to monogenic diabetes, a Mendelian disease, where people that have that variant have a very high likelihood of having the disease. So these are very high impact variants. On the other end of the spectrum are very common variants that all of us will have, that have a very small individual effect on disease. And we know, as I'll discuss, hundreds and thousands contribute to complex diseases like type 2 diabetes. So monogenic diabetes can be very helpful, I think, as an initial way of conceptualizing how genetics can inform on endotype. So the idea is that we can have a gene that informs on disease mechanism, and then the dream is that it informs on treatment. And to some extent, we see this with existing subtypes of monogenic diabetes. So there's the subtypes related to maturity onset diabetes of the young, or MODY, where we have genes such as GCK, HNF1 alpha, and HNF4 alpha that have specific functions within the pancreatic beta cell. And based on the effect that they have on the pancreatic beta cell, that informs treatment. And I'll take you through a little bit more detail of one example there. And then another example is the gene PPAR gamma that causes in adipocytes impaired, so in fat cells, impaired fat differentiation, and causes autosomal dominant partial lipodystrophy. And we know that that gene can be targeted for treatment. And what's really cool in that example, and actually also for HNF1 alpha and HNF4 alpha, is that the same medications that can treat those kind of rarer subtypes of disease also work for all people with type 2 diabetes. So the concept that I want to get across here is that you have a distinct genetic pathway that leads to diabetes and informs management. So if we try to really unpack that and take an example with HNF1 alpha, what we're saying is that you can start with a rare pathogenic variant, and we know the gene, we know the predominant cell that is acting that's relevant to diabetes. And where I see the endotype coming in is the specific dysfunction that we see. So beta cell dysfunction that's impairing insulin secretion that leads to diabetes. And then the idea that we have, the kind of the dream is that then you really can be specific in targeting that endotype or that dysfunction when you treat these patients. So in the case of HNF1 alpha monogenic diabetes, or MODY, we know that because it's a dysfunction in insulin secretion, if we give these individuals treatments that promote insulin secretion, that we can treat their diabetes. So sulfonylureas have been, historically the treatment of choice, and then more recently, there's a lot of recognition that GLP-1 receptor agonists, likewise with their improving insulin secretion, can be effective therapy as well. And then in particular, and this is very profound in these patients, because often they'll be on insulin at the time of diagnosis, and by treating the actual underlying issue, you can put them on you can take them off of insulin and give them an easier management regimen. So monogenic diabetes can inform multiple different endotypes. And one way of thinking about this is kind of borrowing from cancer, where there's a concept of hallmarks of disease, that we can start to think about hallmarks or pathways of diabetes. And these will often, be thought of in terms of pathways of insulin resistance or insulin deficiency. And we can start to fill these in from known monogenic conditions. And I'll just mention as a kind of plug that there's still more genes causing rare forms of diabetes that remain to be discovered. And so there's an initiative through the US government, through NIDDK, the National Institutes of Health, to identify people across the United States with rare and atypical forms of diabetes. So if you yourself or one of your patients has a form of diabetes that you don't know what it is, and it's particularly if it's non-autoimmune, you can direct them to this website, and they can self-enroll in the RADIANT study and get whole genome sequence, and the goal to identify more subtypes of diabetes. Okay, so that's in some ways where you have kind of a more straightforward endotype. So what does this look like in terms of type 2 diabetes, where we're dealing with a more complex disease? So there's been massive genetic discovery for type 2 diabetes, over 500 genetic regions associated with the disease, and these are distinct. And what I'm showing you here is an overview of the discovery over the past about 2 decades. And the publication ID is shown along the bottom by the year. And then you can see these are sample size and thousands of the studies involved. So these genetic association studies involve thousands to now millions of people with diabetes and without. And the concept is that you're looking for particular genetic variants that are associated with disease, so seen at a higher frequency in either people with type 2 diabetes or in controls. And one thing that you can see is that there's a lot of purple, and so purple represents that the European subset of genetic ancestry, the subpopulation of European genetic ancestry contributing to these studies. And it's very well recognized as an issue in the field that there's this bias towards populations of European ancestry in genetic discovery. And so this has been a real effort. And so there is, over time, you can start to see more of the other colors, but still remains a limitation in terms of genetic discovery. So there's been a lot of interest in taking all these genetic variants and putting them together and generating a polygenic score. So you can find people that have a lot of genetic variation that is increasing diabetes risk, and find people that are at the highest genetic risk for this polygenic score. So when you do this, you're kind of combining all genetic variation together across many different pathways. So you're identifying people that have a mix of multiple mechanisms contributing to their diabetes. We've been interested in trying to separate all these genetic variants instead into their distinct disease mechanisms, with the idea that potentially by doing this, we can identify endotypes. So the issue is, why this is actually very challenging, is that all these genetic regions associated with type 2 diabetes, the vast majority have an unknown causal variant that's actually like disease causing, and an unknown gene. So they're often non-protein coding regions of the genome associated with diabetes. So there's been, painstaking effort to go from a particular genetic region to a causal variant, a causal gene, a implicated cell type, but this is very difficult. So we've been interested in looking at ways of kind of jumping from the sets of variants to physiological function or endotype, using associations that are shared for sets of genetic variants with different cardiometabolic traits. So this is what we call like a cluster analysis, a genetic clustering, where we take the genetic loci and try to group them together into shared mechanism. So we had initially proposed an approach to do this in 2018, and we identified 5 salient genetic clusters of type 2 diabetes. One thing that was really reassuring was around the same time we were working on this, completely independent from our work, another effort that used a similar but distinct set of genetic variants and traits and clustering approach converged on these same 5 genetic clusters. So what we were identifying were mechanisms of, so 2 mechanisms of insulin deficiency and 3 mechanisms of insulin resistance. So I'll just mention that since these publications, there's been continued work in the space. So we published about a year ago an expanded effort with over 300 variants and 64 traits. We now have a multi-ancestry analysis that's in medRxives, where we're up to over 600 variants, 110 traits. And then there's an effort that we're also involved in through the Type 2 Diabetes Global Genomics Initiative, where there's also a preprint. And what's really remarkable, I think, is that all of these efforts are converging on these same 5 processes. Now, with more variants, more traits, there's additional processes that are being identified, but we still see quite, robustness that these 5 kind of pillars are continuing to reemerge in these analyses. So because even though these came from, an earlier publication now, I think it's still helpful to talk about these 5 genetic clusters or mechanistic processes, because they have kind of withheld the test of time now in subsequent analyses. So to give you an overview, the 2 genetic clusters that relate to insulin deficiency, so the way that we know they're relevant to insulin deficiency is that the same allele, the same variant that increases type 2 diabetes risk is associated with reduced fasting insulin. And then these 2 clusters just differ in that there's a subset that are associated with increased proinsulin, whereas another subset are associated with decreased proinsulin. So with our clustering approach, we use a soft clustering approach where we can, we actually are clustering both traits and loci. So we can see that, these patterns of key traits that are the most highly weighted or driving traits to generate these clusters. And we also get a set of key genetic loci. And what I've highlighted here is that sometimes there are genetic loci that we recognize, where even though many genetic loci, we have no idea what the causal gene is, sometimes there are those that we do recognize where they're known to be relevant from previous work, or in here from monogenic disease, that we see these loci, and there's been work to show that the common variants in these genes or in these loci relate to these genes. So we're seeing HNF1 alpha, HNF4 alpha that I had mentioned to you earlier is important for insulin secretion. And so we can, putting these traits in the loci together, we can infer a disease mechanism. And I think we can think of these disease mechanisms as endotypes. And then within the space of insulin resistance, we identify 3 mechanisms. One that's kind of the obvious one that we would tend to think of with diabetes. So increased fasting insulin with increased body fat, percent body fat. So markers of adiposity. We see monogenic locus that overlies the monogenic cause of obesity. So it's obesity mediated or adiposity mediated. Another cluster that relates to increased fasting insulin, but decreased body mass index and percent body fat includes well-known gene locus related to the gene for monogenic lipodystrophy. And so this is a fat distribution mediated or lipodystrophy related mechanism. And then finally, we see a mechanism related to liver lipid metabolism. So what I believe we're seeing is that there's this convergence of genetic endotypes of diabetes, where you have in the monogenic space like a real slash of a hammer onto the pathway, whereas in polygenic disease it's more of a poke. So it's, we know that pathway is being implicated, but not to the extent that it's causing as severe of a manifestation of disease. We've also been able to connect these sets of genetic clusters to relevant tissues using epigenomic data. And I'll just mention that, in our most recent work, we've been able to do this at the single cell level. So where we can connect, for example, the beta cell to pancreatic islets in our most recent work, and we also now kind of with more loci, more traits, the beta cell gets separated into 2 subsets, where we see that there's still this enrichment of the pancreatic islets, but we start to see differential enrichment based on particular cells within the pancreas. Okay, so now how can we apply this to people? So the concept is that we can generate a polygenic score using the sets of variants in the different clusters. And what we've shown is that if you take people with diabetes and you know nothing about them clinically, you just have their genetics and you generate a polygenic score, you can take people with the highest score unique to a given cluster, and then look to see clinically what these people look like. And we see very significant differences that relate to the disease mechanism. So for example, we see the two insulin deficiency clusters, individuals with diabetes compared to everybody else with diabetes have lower C-peptide levels indicative of reduced endogenous insulin production. And we see, other patterns that fit the disease mechanism. Although I want to point out that this is, these are analyses involving thousands of people where we subset, to hundreds and look at those hundreds compared to everybody else. And we see these significant differences, but they would never be observed on the individual level because the differences are too small. So I think, they're real, they're just subtle so that you can't see them on an individual level approach. We also see the same concept of significant but small differences in other comorbid metabolic disease risks. So for example, hypertension, coronary disease, renal function. And what's really, I think, really cool here is that these are all genetic variants that are increasing diabetes risk, but have differential effect on these other metabolic diseases. And then finally, an area that I'm really excited about is how we can start to take these endotypes and really start to dive into what the cellular function underlying them is. And so this is work that's being led by Melina Cloud-Sister's lab at MGH in the Broad. And her lab particularly studies fat biology, adipocyte biology. And so she's been very interested in the lipodystrophy polygenic cluster. So this type 2 diabetes cluster that I had described to you, that in this cluster we'd shown in our epigenomic data was most enriched for adipose tissue. So what she's been able to do is take fat biopsies from individuals undergoing abdominal surgeries. And she has samples that are subcutaneous adipocytes as well as visceral. And then her lab can generate adipose tissue-derived mesenchymal stem cells. So she can actually take these cells through development and then perform cell painting, lipocyte painting that can profile the different cellular processes. So for example, the fat cells, the mitochondria, the DNA. And what's really cool is that there's, with a machine learning approach, you can take these images and separate them out into these different processes. And then you can ask the question, if you take these same individuals and stratify them based on polygenic score, would you see differences in their cellular profile? Now, what it turns out is that if you just look at their type 2 diabetes, the full polygenic score, there's very little signal you see in differences of people that are high end of the polygenic score versus low end. But if instead you take the more mechanistic endotype, specific lipodystrophy polygenic score and look at high versus low, you actually see this explosion of signal, and it's particularly within the subcutaneous adipocytes. And what I think is really remarkable is that these differences now are visible at the individual level. So this is a composite of people that are high risk versus low risk. And with the naked eye, you can see differences in the cellular profile. And just to take you through what we're actually seeing, we're seeing smaller green lipid particles. We're seeing more red for mitochondrial function. And what this relates to is actually a similar profile that you get with monogenic lipodystrophy genes. And it's showing that it's indicative of apoptosis of these cells, that you have reduced lipid accumulation in the subcutaneous adipocytes with the lipodystrophy phenotype, because there's the fat is preferentially stored in the viscera as opposed to the subcutaneous tissue. So this fits with very much with our understanding of the endotype, but now we can actually see it in people with that don't have this rare disease, but in the more common population using the polygenic score for lipodystrophy. Okay, so then just to put this together, so what are we really talking about? There's many ways. So Dr. Stefan gave a really beautiful overview of the ways that have been proposed to subclassify people with diabetes. I think the challenge with endotypes in type 2 diabetes is that most likely we have many disease processes relevant to each person. So how do we separate these out, and what does that mean for a given person with diabetes? Mark McCarthy has years ago had proposed this idea of a palette model, that there's many disease processes relevant to a person, and then you see kind of the driving mechanisms for a given person. And this is what he's called a palette model. And I think the way that we can, the vision that I have for this is that there will be some people at the extremes where their disease is driven by a particular mechanism. And those are the more straightforward ones. Those are the people with monogenic diabetes, or extreme of a polygenic process, or extreme of potentially an environmental process. But they're more homogeneous in terms of their disease process or endotype. Many people, though, or most people probably, are going to be a mix of many processes. And in fact, those same processes probably impact not just diabetes, but other metabolic conditions as well. So my personal view of kind of where this is probably going to be the most useful for type 2 diabetes is going to be in terms of developing treatments that we can take these endotypes, start to understand the underlying cellular process, and develop treatments that we then potentially can focus in on people that are most where their diabetes is most driven by a particular endotype. But we may find that these treatments work for many people, as long as there's some substantial contribution of that endotype to their disease. So, but that's hypothesis at this point. But so what do we learn from genetics about endotypes and diabetes? Well, we certainly have clear endotypes illustrated from monogenic diabetes. In type 2 diabetes, I think we're starting to see some nice illustrations of endotypes, and where some overlap with monogenic disease. We can connect these to tissues and even single cells at times, and we see associations with other metabolic outcomes. I think the big limitation for diabetes is that it's complex. There's going to be probably many processes relevant to a given person, and the polygenic scores can stratify people, but we see differences more at a group level than at an individual level. And although I think the cellular level is very exciting, that there you see larger effects, and we're continuing to try to improve upon this issue of genetic diversity. So I see the next steps as starting to build these models to be as we involve more genetic variation, and we look to more complex diseases, and hopefully develop ways to inform drug discovery. So thanks very much. I'm happy to take questions.
Great, thank you. Thank you so much, Dr. Udler. That was a really interesting and informative presentation. I want to make sure that we stay on time. So I think we only have time for one or two questions. So we have a question from Dr. Venkat Narayan, which is actually more of a statement, but we can open it up for discussion. So Dr. Narayan says this, which I'm assuming by this he means looking at the endotypes from a genetic standpoint. This is the better approach, starting from underlying biology and then connecting it to phenotypical presentations. The other way is potentially misleading, and obesity gets in the way. Time to decouple diabetes from obesity to study the etaology. After all, most genes related to diabetes are islet cell genes, not insulin-resistant genes. Are we ignoring glucagon?
Well, so yeah, there's a lot of great thoughts in there. I think we can. I don't think we can ever fully ignore or just focus on the genetics. Like, there's always going to be a component that's very important to disease for the environmental factors and social determinants of health, et cetera. But yeah, I do share the vision that the genetics can be very useful. And I agree with you, glucagon. I think Dr. Stefan had mentioned earlier that our ability to cluster is always going to be dependent on the variables we have. So we're getting better and better GWAS of kind of dynamic insulin measures and more useful biomarkers, but glucagon is one that has not been well captured in GWAS. And yeah, I think that would be fantastic. I do think that's very important, and it's missed right now.
Okay, so for the sake of time, we're going to cut the questions a little bit short today. But again, thank you so much for this really interesting and informative presentations. So we'll now dive into our second hour of the Emory Global Diabetes Research Center Symposium. So the second hour, we will hear from a panel of 4 experts who will discuss their vision for the future of research around diabetes endotypes, as well as clinical and public health applications. So each panelist is going to give a brief 5-minute presentation as to their overall, what we're calling big thoughts about how the field of diabetes endotypes should move forward, given the current evidence that we've heard from Dr. Stefan and Udler in the first half, as well as other evidence out there. And then after each panelist has had their chance to speak, then they will have a chance to reflect on or give a counter argument or counter opinion as to ideas from the other panelists. So I will introduce each panelist one at a time. So first we'll hear from Dr. Anjana from Dr. Mohan's Diabetes Specialties Center and Madras Diabetes Research Foundation in Chennai, India. Dr. Anjana.
Thank you so much, Anjali, and it's a pleasure to be here and discuss these thoughts. So here are my 2 big ideas. The first one is about next-gen AI ML for sex-specific diabetes endotyping and predicting individual therapeutic outcomes from genetic work. And maybe I'll refine that after hearing Dr. Udler's talk right now, and clinical RCT data. So as you probably know, in Asian Indians, we have 4 subtypes of type 2 diabetes. The first and perhaps the most prevalent one is the severe insulin deficiency diabetes, followed by the insulin resistance and obesity related diabetes. Then we have a combined variety which has both insulin deficiency as well as resistance. And the last is the age related. So that is the most prevalent one, followed by the deficiency. So we've also shown that these subtypes have differential responses to microvascular disease, with the insulin deficient types having more of retinopathy and the obesity having more diabetic kidney disease, the combined one having more of both. So what we've done in this is that we've generated an AI ML tool, a web-based tool called as Diana. So Diana stands for Diabetes Novel Subtype Assessment. And what the tool does is that from simple clinical parameters, that when the patient walks into the clinic, we ask age of onset of disease, duration of diabetes, waist, BMI, etc., lipid parameters, and C-peptide is a marker of insulin secretion. We are able to define endotype membership. Now, how does this help us? Now, this is new onset diabetes people are dealing with. So as clinicians, when we're sitting there, it helps us to tell them if they have the better forms of diabetes, whether we can work on reversal with them, or maybe we need more stringent follow-up or aggressive therapy. And this helps the patient as well as the physician to deal with what, and to know what they're looking forward to. So these are the things that we use the endotyping from. We also find, and in this tool, although the AI/ML part looks at clustering for endotype membership, the individual risk of complication is done through logistic regression models for the individual patient. So we have a kind of hybrid thing going on wherein we have clustering on one side, and then you have the individual risk prediction for the individual patient. Also, we have some, thing for the therapeutics, and this is mostly from retrospective data. But currently, we have this clinical trial running, the aggregate study, in which Venkat from Emory is also involved, in which we're looking at the 3 main groups, the insulin resistance, the insulin deficiency, and the combined group, and looking at differential drug responses. And if we have data from this, perhaps we'll be able to populate better in AI/ML prediction. We also saw that there are some sex-specific differences. We don't know too much about this at the current point of time, but we're looking to see whether or not that will make a difference in outcomes or therapeutic changes. So the last part will be, of course, about the genetics. Now, South Asia, the biggest problem we have is we don't have much genetic data, very little GWAS. So looking from there, we would try to see whether we can. I do not know how good we're going to be at this using partition, polygenic scores, etc. But that is the thought behind it. So that explains idea 1. Now, idea 2 is more in the prevention space. And I know other speakers are going to be talking about pre-diabetes and so, but what we thought is that if we're able to generate these polygenic risk scores, or maybe partition polygenic scores, if we wanted the mechanistic pathway, we could identify not just at pre-diabetes, but any time during the person's lifetime, we would be able to target those for behavior change, maybe calculate heavy genetic load, which we do know Asian Indians have, and decide who we need to start treatment for early, who we can wait a little bit more for lifestyle disease. I do not know, but maybe we could come up with something like that in the diabetes prevention space as well. So these are the two big ideas I had, and I'm done before 5 minutes.
Great. Thank you, Dr. Anjana. So hold on one second. Oops. Okay. Thank you again, Dr. Anjana, for your ideas. So we'll now hear from Dr. Omar Bello-Chavolla from the Instituto Nacional de Geriatría in Mexico City, which is in Mexico.
Thank you very much. So following up on some of the discussion that has been brought up by the prior presentations, I am spotlighting two ideas that actually our group has been trying to work with in the past couple of years. The first is actually, I think, a very critical point if we want to move forward with this field, is that we have to come up with a more consistent definition of endotypes. Thus far, a lot of what has been done in many studies has been done with data-driven approaches. So basically, you take a set of variables that could either be clinical or genetics data, and then you try by using different clustering approaches to identify these diabetes subtypes. So I think one of the main problems with this is that it is highly dependent on the specific data set being used, and it diminishes its reproducibility. Some of the work we did in prior years has been trying to come up with a way to sort of improve this approach to facilitate reproducibility of data-driven subtypes. However, it is based, again, on data-driven approaches. So what I'm trying to propose here is we need to come up with diagnostic criteria for endotypes. Of course, the first thing would be to systematize which endotypes we think there are. Dr. Udler already mentioned some. There are the other clinical clusters that have been defined with the Scandinavian groups. And I think the next part in this research area is try to identify either biomarkers or clinical criteria that could better define these subtypes. Now, another part that we have also shown, and also other research has shown, is that many of these, particularly the clinical subtypes, are not stable over time. That is perhaps an advantage of the genetics based criteria. However, one of the things we have to understand is whether the phenotype with which individuals with diabetes start with diabetes, they actually follow through time over time. So I think one of the things that we have to understand is when individuals go from pre-diabetes to diabetes, what is their phenotype? How are they behaving? And whether this phenotype is actually stable over time. I think that is one of the main areas that we have to work with. Perhaps a consensus approach could be ideal, and perhaps this symposium could be a starting point to come up specifically with criteria that could help define which are the endotypes that are the most reproducible in several populations. The other is there is currently sort of a disconnect between clinical and omics-based diabetes endotypes. So we already heard about the genomics-based, some of the clinical phenotypes. However, there is not much work regarding the overlap between them. And that is a problem because there should be overlap. Perhaps we're talking about very similar things, or we're using different things to define them. And as long as we keep talking with different tools about similar things that are with different definitions, we will not be aware of what we're actually trying to define. So of course, there's controversy of whether we even need endotypes, and that will be something that we will discuss, another panelist will discuss from here onwards. But we have to be sure that when we use genomics, when we use clinical data, we're talking about the same thing. And I think that is something we have to understand. Actually, some of the members of my group, particularly my future PhD student, Juan Pablo, is trying to develop a framework to understand how these clinical omics-based diabetes endotypes overlap and whether this is clinically meaningful. And I think that is something that we have to look forward in the future. Thank you very much.
Great, thank you so much. So next we're going to hear from Dr. John Dennis from the University of Exeter in the United Kingdom. Dr.
Great. Hi, everyone. Great to be part of this really interesting session. So the first big idea I want to cover really is, do we need diabetes endotypes at all? And I think, from what we've heard today, already it's clear that we would be great if we could move towards reproducible, distinct diabetes endotypes. And we've heard how research in this area is already starting to inform both etiology and, with a potential roadmap to clinical translation. I think we've also heard from Miriam a really good example of almost a perfect translational approach, which is monogenic diabetes, where we have a distinct genetic etiology that determines both clinical management and treatment requirements. We've also heard that type 2 which is my main area, is a lot more complicated. And we're not really yet at a stage where we have subgroups that determine management, treatment, and so on. And so really, in their absence, I would argue that potentially not enough attention is being paid at the moment to the alternative, which is using the individual features of people with type 2 diabetes to predict their outcomes directly, whether that's progression, treatment response, and so on, and using that as a more pragmatic, actionable precision medicine approach. I would caveat this is broadly my own area of research, this idea of direct outcome prediction. So the key difference is using features to predict outcomes, not assigned to subtypes. Really, this area is a very active area of research, and we've been able to show it's more accurate than the phenotype-based clusters for both predicting treatment response and progression. And we now have good evidence that routine features alter glycemic response and other short-term outcomes for all major type 2 therapies after metformin. And now we're also starting to build evidence that really combining features in what we call treatment selection models can actually support targeted treatment of specific medications to individuals likely to have the best risk-benefit profile. So really, my conclusion here is endotype research is incredibly important, but there are clear short-term gains from considering other approaches as well, in particular individualized prediction modeling. I'd just like to quickly follow on to the second big idea, which is thinking about how we might start to translate and get evidence for clinical translation of a precision medicine approach. For this, I'd like to particularly highlight the TriMaster trial, which was led by my colleague, Bev Shields at Exeter, published in Nature Medicine last year. And this is really one of the first examples of testing prospectively strata defined from data. In this case, subgroups defined by BMI thresholds of 30 and normal reduced kidney function. And really, I think there's a few lessons we should consider as we move forward with endotype-based research and thinking how we might design trials. Firstly, this sort of approach needs a very specific trial design. In TriMaster, we focused on within-person differences in response, looking at 3 major drugs, SGLT2s, DPP-4s, and pioglitazone, with patients randomized over a 12-month period to try all 3 drugs. So this enabled us to look at short-term outcomes such as 12-week HbA1c, but not longer-term outcomes. And reassuringly, we did see differences in drug response that were concordant with our predictions from data. So good evidence for a potential precision medicine approach, but highlighting some challenges, which we need new approaches to design these trials. Sample size is going to be a huge challenge. TriMaster, we needed 500 participants for very simple subgroups, but when we've got less common subtypes, more strata, that sample size is going to go up. And it's really unclear how you can design precision medicine trials using endotypes or other features to look at these longer-term cardiovascular outcomes. Particular considering larger CVD outcome trials, as we know, themselves recruit only high-risk subsets in general and need many thousands of people to show average treatment effects. So really, I guess my big idea is that to push this forward, what we need is high-quality analysis of existing data sources, especially post-hoc analysis of RCTs, as well as these smart implementable trials such as TriMaster to really build this evidence base for endotypes and precision medicine. Thank you.
Thank you, Dr. Dennis. And so next we will hear from Dr. Ram Jagannathan, who is at Emory University in Atlanta, Georgia, United States.
Hey, thanks, Anjali, for a gracious introduction. So the discussion on diabetes endotype is particularly timely and pleased to contribute to this discourse. The first big idea I wish to put forth is the utilization of diabetes phenotype for a comprehensive biological classification of diabetes. So as we all know, type 2 diabetes is complex and poorly understood collection of heterogeneous conditions. The current definition and classification of diabetes are imprecise and rather outdated. Presently, the disease is defined by blood glucose concentration using imprecise and static measures, along with cut points based on predictions of micro and macrovascular complications. Moreover, the regulation of glucose in the circulatory system is not thoroughly understood due to the intricate physiological and pathophysiological processes that work together to maintain glucose balance. So my idea is to propose the concept of diabetes endotype to establish a biological definition of the disease. For instance, various phenotypical clusters of diabetes have been identified, demonstrating differences in pathophysiology and variation in insulin deficiency and insulin resistance, and it differed by race and ethnicity. Our own group's data led by our postdoc Pradeep revealed a novel clustering features, indicating a higher prevalence of severe insulin deficiency phenotype in South Asians living in India with a low body weight. So to advance the precision medicine for diabetes, we must transition toward a biological definition and identify the molecular basis of the disease. So this necessitates the identification of novel biomarkers for diabetes phenotypes, and also taking the inspiration from other fields such as Alzheimer's disease, which has a biological classification system called AD continuum, using ATN classification system. We should develop a similar approach for diabetes classification. So, I hypothesize this system could integrate multimodal biomarkers from various organ systems involved in glucose regulation, including liver, muscle, pancreas, and kidney, and also employ multi-omics data, imaging biomarkers, and novel biomarkers to enhance our biological understanding of diabetes phenotypes. Thereby, it can enhance the therapeutic potential. And my next big idea is to discuss characterizing the diabetes, pre-diabetes endotypes for personalized diabetes prevention. So while most concepts related to endotypes predominantly focus on diabetes, we argue that there is a crucial need to comprehend the heterogeneity of pre-diabetes and identify novel endotypes for personalized diabetes prevention. So diabetes prevention studies in the US and Finland indicate that the onset of type 2 diabetes is preventable by over 50% during the pre-diabetic stage, and also has the long-term benefits. However, studies conducted by our group, led by Mary Beth, have demonstrated that the efficacy of lifestyle intervention is approximately 28% among South Asians. So this underscores the need to characterize pre-diabetes phenotypes in this specific population. Unpublished evidence from our group reveals that over 3/4 of the population exhibit insulin deficient phenotypes. Given that the mechanism of action of most of the lifestyle intervention components aim to improve insulin sensitivity, there is a pressing need to study and characterize pre-diabetes phenotypes, and also it will enable the identification of individuals who can benefit from customized intervention, considering the specific pre-diabetes endotypes prevalent in this population. So also unrelated to this topic, in addition to phenotypic clustering, it will be really interesting to do clustering based on socio-behavioral and environmental factors. So, for example, we have recently finished a manuscript using the longitudinal MESA and handles cohort, and showed that we could classify individuals based on neighborhood characteristics such as aesthetic quality, safety, healthy food, walking environment, and social cohesion, and also based on individual characteristics such as anger, anxiety, chronic burden, lifestyle discrimination, and everyday hassles. And interestingly, some of the adverse neighborhood and individual clusters are significantly associated with individual incident diabetes than the others. So thank you so much for the opportunity, and wish you all the best. Thank you.
Great. Thank you so much, Ram. So thank you to all the panelists for sharing your ideas. And I would now like to invite all of the panelists from our second half to give a brief 5-minute or so, either rebuttal or question, or rebuttal, reflection, or ask questions to the other panelists as to what you've heard so far. And we'll start with Dr. Anjana.
Okay, so thanks, Anjali. I think I'll start with Ram. Ram, I really loved your ideas about clustering environmental phenotypes as well as how do you think something like, anger or stress or something like that would, that's going to be a very, very complicated analysis, Ram. Do you think, it's going to be, I think, even more complicated than the polygenic risk scores? There's like 1,000 variables there.
No, we have used questionnaire-based data, to derive anger, and also, for neighborhood measurements, there are satellite and other data are available, so sensor track data. So we have leveraged that in order to kind of cluster the individual. So I know it was kind of straightforward analysis, to be honest. Yeah. So in fact, we are kind of now analyzing the UK Biobank data, neighborhood deprivation data, to link that with chronic kidney disease. So that's in integrating both phenotype and environmental data. It's something I'm really interested in, so because I know that is the kind of precision medicine, right? So we need to integrate all the available data, what we have. So yeah, thank you.
Thanks, Ram. So I think I'm supposed to be asking the questions now, and people are supposed to be answering later. Is that it, Anjali?
Yeah. So I think that we'll just leave it a little bit of an open session. So I think now I'll leave it to Dr. Bello-Chavolla to ask his question and just kind of keep everything on track. So any questions, reflections, thoughts on what you've heard?
Yes, I have a comment on the question for Dr. Dennis. I know a lot of controversy has come to whether we even need endotypes at all, and I think that is a very valid question, particularly as this field keeps growing. I've seen some comments regarding the use of individual variables for prediction of outcomes, and I think that is very relevant if you try and think from a precision medicine perspective. But I think, and I would argue that regarding the endotypes particularly, when you try and use these endotypes, many clinicians have actually approached me and told me that is something that we have intuitively thought about even before this has been published, right? So there are clinical endotypes, and the idea is whether these are actually useful for outcomes. I think that is a very different question, right? One is a pathophysiological question, and another is an outcome-driven question. And I think those are the two things that perhaps we have to keep in mind when we think about endotypes, right? One is a pathophysiological perspective, and I think that was highlighted by Dr. Udler very, very precisely, particularly when looking at the genetic endotypes, right? And the other is particularly, I think, with clinical variables, which I would agree with, that perhaps using individual clinical variables and their changes over time are more useful for prediction of outcomes than just trying to use these clinical variables to define endotypes. And that is, I think, many of the changes over time that we have modeled in endotypes are actually artificial. And I think that is something that we we have to consider because they are data driven and this changes over time, right? So if the variables change over time, of course, the phenotype appears to change over time, even if the overall health status of the patient is not particularly different. So I think I would like to hear your thoughts on whether you think these endotypes could also be useful for pathophysiology, or whether you think that if they are not proven to be useful for outcomes, they would probably not be that useful. I think that is my main takeaway from your discussion.
Yeah, thanks. I think you've raised some really interesting points. And I think my view is certainly that the genetically defined clusters, I think they have enormous potential to further our understanding of etiology and underlying mechanisms. And I think, Miriam's conclusion was a really nice one. One way that could help would be designing whole completely new therapies with, different mechanisms of action. And I think, there is some confusion in the field to me about separating out, research that is to further etiology from research which is focused on clinical translation, and the most accurate prediction for patients. And to me, I think it's quite clear that the evidence for endotype cluster approaches to date is that they don't outperform individualized models. I mean, that's almost not surprising, as certainly the phenotypic-based models involve essentially assigning individuals to subtypes, so you're losing information, so prediction will suffer, right? I mean, one question that hasn't been fully answered yet is to what degree could the actual clusters, enhance prediction over individualized models? So do they capture some sort of higher level heterogeneity that's missed when you just, use a regression-based approach to sort of predict progression or treatment outcomes? And, that's one way they could have utility. I haven't seen that answered robustly yet. But yeah, I think my conclusion really is, I think setting up the research, what exactly we want to achieve by these sort of endotype-based research is really important at the start, and etiology definitely huge potential. I think clinical translation, especially in the near term, I think there's a bigger question mark over that, and we should at least be considering alternative approaches that we know have worked well in other diseases, cardiovascular medicine and risk prediction, that's really well established. Can we bring that sort of thing approach over to diabetes more generally?
Great. Thank you, Dr. Dennis. Do you have any thoughts or questions for anybody else on the panel?
Yeah, I think my major comment, I think this was picked up by a few of the other panelists, is, I think something we really need to consider early on and now is, health equity. When we think about endotype research, ensuring diverse populations are involved, in model development, from an early on, assessing how well these proposed strategies work across different populations. I mean, the reason is, there's huge, risk of making things less equitable if you have models tailored for one population that don't do as well in other groups. So I was interested in people's, opinion and insight into that area. And I mean, the other thing that I guess is interesting, that what we try and do really is prioritize low-cost routine clinical features that are tested anyway when developing models for diabetes heterogeneity. The idea is then that low-cost to implement worldwide, right? So potentially a solution. I think the danger is, it's very tempting to take the latest technology, measure everything under the sun in patients and come up with new strategies and find new groups and things. But to me, there's that becomes farther and further. The danger of that sort of research becomes further and further away from clinical reality. So I'd be interested in any other of the panelists' views on that sort of trade-off as well. But yeah, no one specific. Anyone who how we define endotypes, I guess. Yeah.
Okay, I think you brought up a very important point, and I think given the very diverse set of panelists we have today, something that we have to highlight. There is a need, if we're going to do this, if we're going to define endotypes a lot of the research that has been done in many diseases has been primarily done in either European or Caucasian populations. And that is very limiting, particularly when we consider that there are very ethnic-specific traits of diabetes, as Dr. Anjana has highlighted, particularly for subcontinental India, right? And if we look at Mexicans, for example, there's also very different traits. For example, how adiposity impacts diabetes risk is very different in Mexicans as it is in other populations. So I think if we're going to do this, we have to start exploiting all of the data sets that we have available, for example, in genomics research, trying to expand upon the phenotypes and try to see if these phenotypes, particularly from an etiologic point of view, are very similar across ethnicities, or if there are ethnic-specific differences that condition etiologic differences in diabetes that may influence either treatment selection or outcome prediction, because I think that would become very relevant if we're trying to individualize diabetes treatment before jumping all the way onto the individual, which I think is very ambitious and perhaps a bit of a high reach at this point. We have to start at least considering ethnic-based differences, which I think have become very obvious. And I think the second point that you raised is very interesting. And that is something that we actually touched upon in our research, which is the clusters as they currently exist are not feasible to be applied in many, many populations. And that limits our understanding of how these endotypes actually may work in different populations. Unfortunately, because, for example, measuring proinsulin, measuring adiponectin is very unfeasible in many of these settings. Trying to find population-based data sets which are large and rich enough to be able to not only provide a representation of diabetes heterogeneity, but a nationally representative one in many of these countries, I think it will be most challenging. So I think it is something that we have to strive for, perhaps, and again, using these as a platform to touch base with many experts in the field. We have to promote research in minorities, and we have to promote research which is affordable so that we can actually hope for clinical translation in this. As it currently stands, I think it's very hard to think of translation, and it's something that we have to fill the gap in.
Thank you. So Dr. Anjana, I know that you didn't get much of a chance early on to ask multiple questions. So if you have anything additional you'd like to add.
Yeah, well, I was just going to say something about the inequity thing that Dr. Dennis asked about before. It's very, very relevant in our situation. And hence, even when we took the variables for clustering, we had to be very cognizant of the fact that it had to be to be able to be replicated in primary care setting and things like that. So, yes, I think the most important thing is who we're reaching and what we're doing with this. So I'd like to state the example of the MODI genetics that we've done. So we started off with the phenotype. We went all the way up to the genetics. And today in the clinic, we are able to offer low-cost gene panels to, offer MODI genetics. It's happening right now. So I think we have to start big, and it always starts at one point. But I think over time, this is then condensed into a form that is available and affordable to the common person. And so I think in Asia, and I can maybe generalize for South Asia in general, we're still a very long way off. But I think we have to start thinking towards it and moving towards it. And slowly, I do agree, we do need things that are backing our phenotypic. That's the only thing we have as clinicians. And Dr. Chavolla was saying that when we made these, divisions, clinicians were instinctively doing this anyway, because they are, after all, phenotypic characteristics, right? So what we do need is some gene backing for this, and that has to be in an affordable space over time. And I think it will come if enough number of people are working towards it. And I think that commitment of being inclusive about all ethnicities is something that's very important. So that's one thing. And Anjali, I would I did like to ask Dr. Chavolla, sorry, I do not know if I said your name right. When you say point of care testing, that's another thing that becomes very relevant for us again, because number one, standardization becomes an issue. And let's say there's bias over bias once we start using these devices for whatever purpose. And especially in the diabetes space, whether you're looking at A1C or whatever else you're looking at, we still have issues. And second, it's way more expensive and not freely available. So of course, we would love, it's easier to do, and we'd love to move towards that, but we find a significant hurdle for these reasons. So do you have any comments on that?
Yeah, thank you very much. I think when we think of the approaches that we could undertake to translate this, I saw a comment in the Q&A where they are saying, if we're thinking of application, are we not limiting how we're progressing? I think it goes hand in hand in one way, because we have to keep in mind the ultimate goal, which is we're dealing with a disease which affects millions of people, of whom we want to improve the lives of, right? So we have to come up with ways to make these advances in science that we're making, also considering the potential applications that we have. So the point of care testing that I'm suggesting, of course, it's way far into the future. First, we have to agree upon which clusters there are, right? If there are, of course, we have the intuitive insights from clinicians, and we have to see if, from a population perspective, they are actually reproducible. I think that is the first point. Then, of course, if the best way to identify them is to measure, I don't know, adiponectin or proinsulin or leptin levels, then those would probably become the mainstay ways to identify them. So point of care, of course, would be very further down the line. I think it is something that we have to just consider, because we have to think of practicality, right? If these phenotypes make differences for outcome, I think the most consistent thing we have to do as scientists is to translate them all the way into the clinic, right? Because there have been many efforts that have been done in several diseases where you see, for example, precision medicine for cancer, right? And you have to measure these genetic biomarkers that are very hard to apply as we're talking about health inequities in contexts like Mexico or in many other countries which have socio-demographic disadvantages. So what is the point of identifying all of this if we do not find a way to make them affordable and to translate them into clinical practice? I think that is something that we have to keep in mind. Of course, I do agree that there are several hurdles that come with point of care testing. And I don't think because we don't even know which phenotypes are reproducible yet, I think we're very far down the line to considering point of care testing, but we have to keep it in mind. I think it's something that we have to keep in the back of our minds. If we find that these are reproducible, if we find biomarkers, how can we make them affordable? Because I think that is something that we have to do in the future.
Thank you. So Ram, I'll turn it to you for any last thoughts or reflections before we open it up to audience questions.
So I don't one question for Dr. Anjana, so I know I'm interested to know, how you are proposing the intervention studies based on polygenic risk score, so I don't.
So the intervention study was not based on the polygenic risk score. The trial was on it was on drug response. Okay. So yeah, we took the three main groups, that is the insulin deficient group, the obese group, and the combined group. And just these three, not the age-related one. And we gave different drug. So you have the control as metformin, which is standard of care. But the hypothesis was that that's not really the standard of care for every subgroup, is it? So we've tried DPP-4s, we've tried SGLT2s, and in the combined group, we've started combination therapy. So, and then of course you do have the rescue criteria coming in as well. So that would define whether or not, so metformin is not essentially the drug of choice in every group, right? If you have an insulin deficient variety, metformin is not going to do much at all. Or if it's a combined group, then there's maybe more that we have to do. So I think that is the hypothesis behind it. We're about halfway through the trial right now, and we're hoping to have some results soon.
Okay, thank you. So now we're going to open it up for audience questions for the entire set of panelists, both from hours 1 and 2. So if you have any questions, I know that Dr. Udler didn't get to answer a lot of the questions in the first half. So if you had typed a question for her and it's disappeared and you'd still like to ask it, please type it again. So the first question is it's from Tim Mori, and it's addressed to Dr. Dennis. So you argue that going forward, we need high quality analyses from existing data sources, in particular from RTCs. Where do you see the role of real world evidence to understand the heterogeneity of treatment effects?
Thanks, Tim. That's a really good question. Yeah, quite close to my heart. A lot of our work has involved using observational UK data, almost as discovery research data sets. So looking for things that potentially modify treatment outcomes, clinical features that potentially modify treatment outcomes, then taking what we find forward and seeing how reproducible they are in other data sets, particular RCTs. So that's quite a powerful model, I think, for developing quite a pragmatic approach to precision medicine based on what drugs are already being prescribed in practice. So, you can only model real world prescribing, so you're going to lag a bit behind, tirzepatide and everything coming on. The advantages are, you've got an unselected patient population. These are the people being treated. No selection like we know there's in trials. Of course, you've got major confounding and bias to consider. That's why we like this two-step approach. And, to understand this more, I think testing out endotype precision approaches, whatever kind, in as many observational real-world data sets as possible is, a really important thing, and something we could be doing better and more systematically to really iron out what is true heterogeneity and what is potentially things we're picking up in real world registries data sets that actually it may be more related to unmeasured factors. But yeah, I think both evidence really important and can complement each other if used correctly and carefully.
Okay, thank you. So we have a comment and a question from Dr. Narayan. I think it's directed to anyone who'd like to answer. So when Einstein defined his theory of general relativity, he was not thinking of its application. By thinking always of clinical applications, are we limiting the progress of science? Anybody can go ahead and answer.
I don't think anyone wants to answer that. Venkat, science has to have some final outcome or output, right? The human being at the end is always something that I think we should consider. Doing science for the sake of science is what? Where's the end point, right? So I disagree. We should have something that has clinical relevance at the end.
Okay, well, Venkat says discovery is serendipity.
I saw that. And he can go on like that. So
it's possible to have both in parallel, right? We have to be pragmatic. We have to, deal with the person in front of us in clinic and make our best decisions that we don't have full information. But then maybe, in parallel, we're trying to learn more and then apply whatever we learn but I agree with Dr. Anjana that as we're learning, we always need to keep in mind what's the purpose. With endotype, and we've discussed this a lot, what's the purpose of an endotype ultimately, or subtype? Ultimately, it's to improve either treatment response, disease complications. So it's, I think it's important to have both in mind.
Okay, so we have another question for Dr. Dennis. Genetic variance plays a significant role in pathophysiology of type 2 diabetes subtypes that evidently seen from many publications that came in recent time. How do you think the outcome-based approach, adding the genetic biomarker data, would change the story of precision medicine?
Yeah, it's a really interesting question, and I guess Miriam may want to comment as well. But yeah, it's a really important open question. We don't know to what degree the genetics will add over routine clinical features. Obviously, the genetics, is from birth. The clinical features emerge across the lifespan. So it may be that, a lot of the the very heterogeneity relevance to genetics is actually evident in the phenotype. But we don't know till we look. And I think you need pretty large scale data sets with genetics, good follow up to answer those questions. But hopefully, we're getting to a point where we can start answering them soon. Miriam, yeah. Anything to add?
I totally agree with you. I think the place where genetics can be helpful is when there's a phenotype that's shared by multiple mechanisms. So I think, the example that comes to mind is the Alquist cluster of the SID, severe insulin deficiency diabetes, where some of those people we know from follow-up work, from other work, that sometimes they're antibody negative type 1, autoimmune diabetes, and sometimes there's other, so there's some heterogeneity in the mechanism there. So potentially, I think, there are going to be places where maybe genetics can inform to almost when you have a clinical phenotype, and maybe it can substratify in a way that's useful. But I agree. I think the overall our genetic instruments are kind of weak. The type 1 polygenic score is an example where it's stronger, but a lot of the endotype related scores that I mentioned to you, they have small effect on a person's phenotype, and so likely also will have small effect on treatment response at this point until we're better able to, I think, develop them more. Yeah.
And if I just may add on that, and I think, as you said, for example, looking for genetic risk scores in specific subgroups. So I think this is what we're doing in clinic every day. So I think we should be more brave of skipping the mean, so the middle group. So we want to divide everybody in a group, and I think that dilutes completely our thinking. So as clinicians, we would never do that. As clinicians, we look, as Mark McCarthy elegantly said, with the palette, we start from the extremes. We do the phenotyping. We look on a progression of glycemia, and we don't take only the snapshot, the first measurement. Some person has an HbA1c very high, the other has a low, just on the onset. The other lost like 20 pounds of weight, the other not. So this is actually a problem that in clinical science or in clinic, we actually do a longitudinal follow-up, even if 2-3 weeks, and then we know whether this person is running in a complete insulin deficient phenotype, whether this is the obese phenotype, the lipodystrophic phenotype. So I think this is something that we should take into account when we go with our endotypes. And the second is we should skip the middle, come from the extremes like we do in single genetics, and then actually we will be much more powerful in predicting the progression in these subgroups.
Okay, so we have a question from an anonymous attendee. Question is, when do we stop? Is there a final subpopulation, or should we keep stratifying phenotyping into thinner and thinner and thinner groups? This is not addressed to anyone in particular, so if anybody has any strong feelings about this, feel free to respond.
If I may, I think that is something that we have to take into account. I don't think if we find that there are, I'm just like making assumptions here. If there are 10 phenotypes, then we have to investigate these 10 phenotypes. If there are 3 phenotypes, then we have to keep these phenotypes. If there are no phenotypes, then we have to stick with no phenotypes, right? I think we don't have to limit ourselves to think, for example, Dr. Udler's work and Dr. Alquist's work have pointed to at least 5 phenotypes, right? That is what appears to be the consensus as of now. However, we are not sure if the clinical phenotypes are the same as the genetic phenotypes, and that is something that we have to see. Perhaps when you overlap them, they become 8 or something. And then picking up from Dr. Dennis's approach, if they are not useful for outcomes, they may be useful for pathophysiology, but then we have to see whether they contribute anything to prediction, right? So I think we have to keep in mind that it has to be parsimonious, of course. So we don't have to like have an explosion of 200 phenotypes of diabetes, because that would be detrimental to science. But we have to keep in mind that, as a comment saw before, some of these findings are not really planned, are serendipitous, but they are based on some data that has come before. And so hopefully we will come to a parsimonious conclusion in this, or to a parsimonious output in this area of research. But if it's not parsimonious, then I don't think there's anything wrong with that, particularly if it's useful.
Okay, so I think with that, we will now conclude our symposium. So I'd like to once again give a huge thank you to all of our panelists for taking the time to be with us today and to share their insights. So thank you so much, everyone. We really appreciate all of the knowledge that you've shared with us today. And so just an overall summary. So we've learned today from our panelists that there are many ways to define endotypes or phenotypes. And the work done by the researchers here have shown that beyond the endotypes and beyond what we already know, there are some unresolved challenges. We've learned that clinical translation is important through trials and diagnostic tests. And we've also learned that it's important to understand the pathophysiology of endotypes through basic science research with a focus on genetics, epigenetics, and environment. And so we'd love to hear from all of you as we host other seminars in this area. Once again, the symposium today has been recorded, and we'll be sure to send it out to all of our attendees. Please feel free to reach out if you have any other questions or thoughts for us. And again, we would really like to thank our panelists as well as our audience for being here with us today. So thank you so much, everyone, for your time and attention.
Thank you.
Thank you.
Thank you.
Bye.